All right, that's 10:05, so I think we can start off here. Oh, okay, we still have people coming in, which is quite good. Okay, right, now we've got the people in. Okay, so hello everyone. Welcome. I'd like to welcome you all to the first day of the hackathon, which is, the workshop. And this is being held as the second and last joint event of the year between RSG Australia and RSG-Türkiye, which are both regional student groups that are part of the International Society for Computational Biologists Student Council. So the mission of the ISCB, which is the International Society of Computational Biologists Student Council, is to promote development of the next generation of computational biologists and bioinformaticians. And we achieve our goal through provision of scientific events such as this one and the one we had before and so forth, networking opportunities, training, and workshops, providing educational resources and career advice while attempting to influence policy processes affecting science and education. And my name is Kuray Chawatama. I'm a molecular biology and genetics undergraduate student at Bursa Uludağ University. So merhaba to everyone. And I'm part of the outreach team of RSG-Türkiye, which facilitates the coordination and of joint events with our international counterparts, such as RSG Australia and RSG Iran, I think earlier in the year. And I'm going to be the chair of the Turkish side of this event. So I think just as a reminder, those of you who are not already part of the RSG, feel free to let us know in the chat, and one of our moderators will get into communication with you and let you know how you could join, or play a role, or just passively benefit by being the first to know about us when we organize events such as this one in the future. Good. We still see people coming through. Okay. But without further ado, I'd like to introduce you to the man who you've all come to learn from today. It's certainly not me. And he will also be one of the judges in tomorrow and the day after's hackathon. So he's a bioinformatician, next generation sequencing data analyst, and a research scientist with 5+ years of experience developing both front and back end aspects of a wide range of bioinformatics web tools, as well as handling next generation sequencing experimental design and data quality control. He's currently a PhD candidate at Süleyman Demirel University and a research scientist at the German Cancer Research Center. So for those of you who don't know what that is, it's actually a very big deal. So it's the largest biomedical research institute in Germany, with over 100 divisions and research groups investigating the mechanisms of cancer, identifying cancer risk factors, and developing novel approaches to make tumors and tumor diagnosis more precise and the treatment of cancer patients more successful. And in fact, they've actually had 2 Nobel Prize winners in the last, I believe, 15 years. One for the discovery of human papillomaviruses, which I'm sure you will all recognize as the main causative agent of cervical cancer. And the other one for major advancements in ultra-high resolution fluorescence microscopy. So we certainly wouldn't have like all of the immunoblotting and everything we do in the lab without the guys at this famed institute. So we're definitely lucky to have someone from such a prestigious place to take his time and to come and take us through today's single. Thank you for taking your time to help us out today. I've already given you a pretty heavy introduction, but I'm sure the participants would like to hear it from you as well. Maybe a bit more background on your work at the German Cancer Research Center.
Thanks, Cry, for this nice introduction, and thank you for having me today in the workshop. You can hear me, right?
Yes.
Okay, sorry for my voice at the beginning. I caught a cold and some flu, and my voice can be a bit strange.
Yeah.
And I'm working in the German Cancer Research Center Frankfurt unit as the bioinformatics scientist, and we are generally handling the spatial and single-cell data to create different kind of tools, and such as currently we are working on the donor deconvolution algorithm on the multiomics data by using single-cell multiomics data.
Okay.
Then also we are applying to several different kind of stem cell niche component analysis and the tumor microenvironment data.
Okay.
My background is basically it was all started on the genomics analysis and Intron Healthcare based in Izmir. And I did
Okay.
variant
Pretty big
calling.
jump, I think, from
Exactly.
genomics to transcriptomics.
Exactly. Then I actually work on the variant calling algorithms and the Sanger sequencing, deal with this kind of data. Then I went to Oxford Genetics for a year and worked with the genetics data, then created several web-based tools. Then at the same time, I was doing my master and PhD. Then I started in the German Cancer Research Center nearly like 2.5 years ago.
Wow. Okay.
Happy to be here. And today we're going to follow some single cell stuff, some basic workflows of the single cell. Then you expect to analyze your own data after finishing this workshop.
Yeah, I mean, that's definitely the goal. I think those that will be participating in the hackathon have a much tougher job of making sure they listen and understand everything, because it's going to be quite a tight competition from what we've been seeing. So hopefully you all listen close and feel free to ask any questions in the chat, or directly raise your hand, and Yavuz should be able to answer whatever questions you may have. And hopefully everyone has managed to also install R&R Studio as well as the Seurat library to be able to follow along with the workshop. Was there a R Markdown file you wanted them to download, maybe off of GitHub? We could also plug that link into the chat as well.
I can add it. I can add it to Cry
Okay then.
channel. Then
Right, so you
also
can just send it into the Slack, and I think we can always forward
Okay.
it to the appropriate links while you can do your introduction.
Then maybe that you can also ask that if you already asked, but that if it's smoothly work for everyone else, for the R Studio installing, Seurat installing, R installing, or is there anyone who are having problems?
All right, I'll take the silence as a no. Everyone has R and R Studio as well as Seurat and the data installed. Hopefully you didn't have too many problems doing that. So yeah, I think we can definitely start. Oh, someone here says she's not too sure about the libraries. Esma Nur. So I think I'll get in contact with her and send her the code regarding the installation of Seurat. But other than that, I think we should definitely give it a shot and get started.
Okay. Let's first go with the theoretical background of the single-cell RNA sequencing data and the analysis steps. Just a second. So guys, you can see this page, right?
Yes, we can.
Now, as you know, like it's a joint program by joint hackathon and workshop by our side, RSG-Türkiye and RSG-Australia. Then I'm gonna actually move forward with the current best practices from the Ocean from Australia side, which are actually quite nice presentation. Then after we finish this theoretical background part, we're gonna follow with the practical workshop, hands-on practical workshop, and we can actually interactively work on that part. So let's first start by understanding the difference between bulk data and the single cell data, guys. And we generally heard about the RNA-seq data, or the other name of the RNA-seq data is the bulk RNA-seq data. In the bulk RNA-seq data, we basically take the tissue, then sequence all the RNAs in that tissue by not looking at the particular cells inside that tissue. We just dissociate it, then we just sequencing it. There is no any specific information about the single cells, like in a tissue, like there could be several different kind of the cell types. It could be NK cell or epithelial or different kind of epithelial, stem cells, cancer cell, whatever. But when you're sequencing it in a bulk way, you cannot actually separate them well, because at the end you will have a count matrix and you see the all information in all together. But in the single cell data, we dissociate these tissues into the individual cells from that tissue. Then we sequence them separately. So we can actually know the expression profile of each cell separately. It's actually helping us a lot to understand where is this variability in the bulk RNA-seq is coming from. And is it coming from endothelial cells, from cancer cell, or whatever? Because as I said, in the tissue, it could be cancer and other stuff and other stuff. It's all together. We cannot separate them. But in single cell, we can do this because we dissociate this tissue into the single cells. Then I want to mention about the most popular platform for the single cell sequencing called droplet-based platforms, which are also famous with the reputed from the 10x genomics platforms. In that platform, we dissociate these tissues into the cells, then we put these cells into the droplets, then we add the beads, barcoded beads, then we sequence them, then after that, we can see the transcription profile of each cell. There are also different kind of the single-cell RNA-seq experiments. For the experimental design, there are several different platforms like the platelet base or a lot different actually the platforms. But the most common and most popular one is the 10x one, 10x genomics. So also in our workshop, we're gonna share this, but we will first start with the raw data processing. We will mention about it, then the data preprocessing and downstream analysis. We will mainly focus on that part. So what to expect from a single cell data or an RNA-seq data? Let's look at these three different bars. It's all different genes. Like let's call the first one is gene X, gene Y, and gene Z. We at the end counts the number of the reads, UMI counts from that gene. So at the end, for this particular cell, we have a count matrix, guys. This is only from one cell. We have the counts, which means that the transcripts in the biological way, we have 3 transcripts for gene X and 3 for gene Y and 2 for gene Z. So we can say that gene X is highly expressed than the gene Z, right? So at the end of the single cell experiments, we have clearly much more than the one cell, sometimes more than 10,000, sometimes more than 1,000,000. So for the actually genes, we're gonna call them as features in the next stage. For the rows, we will have the gene names. And for the columns, we're gonna have the samples, cell IDs, cell barcodes. So they're gonna be something like 10,000 barcodes, columns, and maybe the 30,000 of the rows at the end. This is called count matrix, guys. This is our actually most important data formats in this analysis. So as we mentioned, let's first do the experimental design when we start this kind of analysis, taking sample, tissue dissociation, putting into droplets, and the other experimental parts, library preparation, sequencing, having the FASTQ, then we align them to a reference genome. This is, if you are familiar with the any kind of the bioinformatics. This is one of the most common stuff in the bioinformatics, aligning the raw reads that coming from samples into the reference genome. For that purpose, we generally use the cell ranger. It's from the 10x Genomics. It's halfway semi-open source to the tool. So, it's actually a star-based aligner. If you are familiar with Bowtie or the BAA, the cell ranger is actually doing the same thing, but at the end, it creates the bound file and also the count matrix and also some useful metrics. It's star-based aligner, but it's a bit slow. Then you can use the Star Solo or LMFry, which is actually isoform awaited aligner from the developer of the Callisto and Salmon. So we will not further investigate these tools in this workshop because it takes a lot of time and we don't have the FASTQ files, but just keep in mind that if you have the FASTQ file at some point, you will search for Cell Ranger, Star Solar, LMFry. These are the most common tools for the alignment of the FASTQ file into the bound file and to create the count matrix. So here we see a real single-cell RNA-seq count matrix. As I mentioned, this is one of the most important data format in this workshop, in this kind of analysis. As we previously mentioned, the gene IDs, we will call them features. Gene IDs will be the rows in this count matrix. And the cell barcodes, cell IDs, the single cell will be the one column at the start point of this analysis. This is the count matrix. So let's investigate it. For the second cell, this cell, as you see, these barcodes are different for each cell. For this cell, nearly all of the genes have 0 counts, which means that this gene is not expressing, this cell is not expressing these genes. This is, of course, the first interpretation, but like that, there are several technical limitations as well. Maybe there is not enough the coverage for this cell, so it could be 0 for this reason because of this. But let's just interpret it like this. This cell is not expressing these genes, but this cell is expressing NPAL3 gene, and it has 1 counts for them. Let's look at the third cell. Also, it has lots of 0, but it has 2 counts for FGR. So it's actually expressing a lot of FGR. 2 is a nice count for single cell. So it's very high dimensional, guys, as you may see here. We just show the 4 columns here. But generally, typically, we have more than 10,000 cells. So 20,000 or 30,000, 35,000 of the rows. It's very high dimensional, and we have to do some dimension reduction. So we're going to talk about this. But it's also very sparse data. Sparse means that it has a lot zeros like this. So this is the one of the overview of the pre-processing parts, which we will follow in the workshop, practical workshop. We will start with the quality control. We will normalize. We will select the highly variable genes. Then we're gonna check more. So in the quality control step, there are several metrics that we have to take care about, and such as like we mentioned that we dissociate the tissues into the cells, and we are putting these cells into the droplets. But sometimes 2 or more cells can go into the same droplet. So in this way, we actually sequence 2 or more cells as the one cell's output. So actually, it's the mixing two cells transcription profile into one, which is actually quite bad because it can just mix a lot in the downstream analysis. It can have the natural killer cells expression profile and also the epithelial cells expression profile. So we have to actually discard these doublets. For this purpose, there are several tools are developed by looking at their transcriptome or by their variant data information. Or as said, a droplet could be empty, or it could be a dead cell, which has a lot mitochondrial count, or it could be other issues like cell is damaged or dying or no barcodes. So the one other important metrics in the quality control is the mitochondrial count. So if a cell has a lot of mitochondrial count, it might indicate that this cell is dying because of having too much mitochondrial gene counts. But sometimes this information is the general one, but there are, of course, like some outlier informations, like some cell types has higher mitochondrial percentage, such as the epithelial cells. So I always recommend you to check different the thresholds for the mitochondrial percentage. But you can look at the elbow, then here it's around like 10 to 15%. Then we will discard the mitochondrial percentage having more than 15% in the cells. So if it has a lot mitochondrial counts, it might indicate that this cell is dying. So let's discard them, exclude them. Or as we mentioned, the duplet section, this is the other quality control metric. We can remove the doublets by using different kind of tools like duplet finder or cellbender or whatever you want to use. So after the quality control section, we follow the other pair processing part, which is called feature selection. In this feature extraction part, we focus on the highly variable genes, guys. Highly variable genes means that in a single cell data, as we showed previously, in a count matrix, there could be 30,000 genes around across the cells. So most of the cells actually are not that variable between the cells because like there are housekeeping genes or this kind of genes that are quite constant expression profile across the cells, which are mainly the responsible for the construction and maintenance of the cells, etc. So this kind of genes are not explaining the data a lot. But in the later stage, there's a very important part. We will call them clustering. We will cluster cells according to their expression profile. So we have to find the most variable genes. So in that step, we look at the highly variable genes. I'm going to talk more about this step in the practical workshop. So then we will normalize the data because every cell may have different technical background as well, because we can integrate the different data sets, or there could be several different effects or PCR or whatever. So a cell could have a different coverage per genes, but when we analyze them for the clustering, PSA, etc., this gene is going to be like the outliers. Then we have to normalize them to stabilize the variance. So we continue with the normalization step. Normalization means actually like it's an ancient Greek word called norm. which means that line. So we are actually trying to put the constant line and just trying to stabilize these outliers into this line in this normalization step. So if we integrate different data sets, we can do batch effect correction by using different tools like MNN from Lada Hackbirdies or Strategies Lab CCA, or quite common one is the Harmony as well, working in the PSAID data. So we also have to reduce the dimension, guys, because we said it's very highly dimensional data. It could be several columns and several other rows. Then we have to reduce this dimension just to understand the similar cluster of cells. Then for this purpose, we are using principal component analysis. And it's compressing these cells into linear combinations of the genes that captures the variance among the data. But of course, the PCA is not the perfect solution for summarizing the most of this information. For this purpose, we're going to use another tools, which is called UMAP or TCNA. So for the data processing part, we will use Seurat. And Seurat is actually doing all these downstream analysis like highly variable genes, normalizations, PCAs, etc. It's all integrated into this framework, which is called Seurat for the downstream analysis. So we first start with the quality control. Then we will go with the normalization, feature selection, dimension reduction, clustering, visualization, and differential expression. So also let's talk about the UMAP. UMAP is another popular technique, which is nonlinear dimension reduction method. So it's the PCA is a linear dimension reduction method, but this is a nonlinear dimension reduction method, and it's actually preserving the local and global structures very well. So it's very fast. It's of course it has drawbacks, but I will talk about this more in the practical workshop. So yeah, in the downstream analysis, we will do the clustering. So we will focus this in the practical workshop a lot, but let's just look at this data. So in this UMAP plot, we see different clusters. Each cluster shows a different expression profile, guys. So it could be different cell types or cell states, such as this cluster 3 could be the cancer cells and the cluster 2 could be the normal cells. You can think about it like this, but probably here we see different cell types, like cluster 3 could be nature killers, cluster 2 could be macrophages, cluster 4 could be epithelial cells, cluster 0 is another one, blah, blah. Then we can see we can see these cells in the lower dimension embedding, and each dot in this plot is a cell. So it could be 10,000 cells here or 5,000, we don't know, but each dot is a cell. And we are just looking at actually the similarity of these cells into the other one, because it's clustered together, right? Inside this cluster 0, all of the cells has similar expression profiles. Here another one is the same idea. So for the clustering, we can use Lewine or Leiden algorithm to cluster the cells by looking at the shares that are neighbored. I prefer the Leiden algorithm because it's faster and an improved one. Then, by looking at the expression profile of these clusters, the upregulated and downregulated genes per these clusters, we can annotate them, which means that we label them as their cell types, such as these clusters. We look at their expression profile, and some genes, like let's say CD79, was expressed a lot in that cells, and we call them B cells. We label them as B cells. Of course, we check much more genes, markers for this purpose. Also, we can do it automatically by using different tools like Azimuth, Singular, or Cell Typist, if you are working with ScanPy. Here also another the visualization. Also, there's another test which is called proportion test. In the proportion test, we try to understand if the cell types between two conditions has been changed significantly. So here we have, for example, two different conditions. And for each condition, we made a single cell experiment and single cell analysis. Then we do all the downstream analysis and we annotate the cell types. Then we look at the changes in the proportion of these cells to understand if it's significantly changed between the conditions or not. This is called proportion test or differential abundance test or differential compositions test. It's looking at the compositions of cells between the type of tissues or conditions. Then one of the most important tests is a differential expression. So in this differential expression analysis, we try to understand which genes are particularly upregulated in one specific cluster or downregulated. We can use different kind of tests like Wilcoxon, t-test, Walt, or there are lots different tests for this purpose. So let's visualize some differential expression genes. So this is for example, from cluster one. We made the differential expression genes for the cluster one against to all cells in that data. Then we noticed that these genes are particularly upregulated or downregulated inside this cluster. For example, let's look at the top one, CD8A. Let's look at the p-value. It's it's a very significant one by looking at the p-value. And also, we made the multiple testing for the multiple testing correction with could be Benjamin Hochberg. So also it's quite high. Then we can look at the log fold change, which means that how many times it's bigger than the other clusters, fold change, but in the log way, log transform fold changed. So we can see it like the 3 times. And also percentage of these highly upregulated genes in that clusters, which are like the 79% of these clusters, the cells in that clusters are expressing this gene highly significantly with very high log fold change. Then we can input the differential expression data into pathway analysis, gene set enrichment analysis, Go analysis to understand if these highly expressed upregulated or downregulated genes do some particular the biologically important the tasks in the cell. And then we can do the trajectory inference analysis, which is actually creating a trajectory between the clusters. It's fitting cells into those at a time. So you can actually track the developmental stage of your cells or the lineage. So it could be developmental stage, but also it could be just to like you can just subcluster your main clusters to show the fine-grained the subclusters. Then you can see the transformation of your subclusters from one to another one. So the third time analysis is also quite important. Then we can do the cell-cell interaction analysis. Guys, cells are not independently working. These are inside the tissues, and these are actually sending message to each other. These messages are the ligands, receptors. These are markers. So we can actually do the cell-cell interaction analysis to understand which cells are sending which markers, which signals, and which cells are receiving which markers, which signals. We can use cell phone DB in R, or we can use, I would also recommend this one, Liana in Python environment. So thank you for listening to the theoretical part, guys. So does anyone have some question about this part?
As a matter of fact, I do. So first of all, what would be your clustering algorithm of choice, TSNE or UMAP, and why? Beyond just the obvious speed, advantages that UMAP offers.
Yeah, the definitely most important one is it's faster. It's quite faster, maybe 10 or 15 times faster. And if you have a big data, actually, this is very important, right?
Okay then. And how important would it be for us to actually understand intrinsically how these algorithms work? Do you think bare minimum understanding a surface level such that we can use it is enough? Or in your time as a data analyst, has it ever been useful to know exactly where the algorithm might be going wrong if something is to pop up?
Actually, this is a very nice question, right? And really, like this is very important because there are several different kind of tools and different kind of the mathematical formulas behind them. So if you really try to understand all of them, it's really time consuming, and you cannot do this because single cell field is the driver in the transcriptomics, and every day the peoples are publishing a lot of tools. So I think minimal understanding is important for the UMAP, but for the differential expression part, this test characteristic, and for the PCA, and for the highly variable genes, and for normalization, I would strongly actually suggest people to learn what is this downstream analysis steps and what the people are using them, why they are using them, because it's important. Like when you look at the UMAP plot, for example, you can try to just interpret it like if the clusters are like the closer to each other, they might be similar clusters, right? Interpret it like this, but it's not correct because UMAP distances in the UMAP is actually not telling us a lot.
True.
Preserving the local structures much better. So in the PCA analysis, yes, these distance are important.
True.
It's also not preserving the global structure. Then it's important to know this kind of information, I think. But other than this, I think you don't need to know this mathematics behind this, like the UMAP or TSNA and this kind of stuff. But I will also share some more information in our practical workshop.
All right, all right. We definitely appreciate that, and I think this was quite relieving since historically biology students tend to struggle with math, and I think that's why we see a very low turn up of people being interested in bioinformatics. But I think once that barrier, once people understand that that barrier of math really isn't there, we should see more people falling in love with the subject. And then I'd like to bring one from the chat here. I had answered it myself, but would want to know your opinion on it. So there was one here asking about, the housekeeping genes and the fact that they don't change from data set to data set, or sample collection to sample collection. And I had responded that typically we use very specific data sets. For example, like you can download it, it's a library, and it contains all the housekeeping genes you're to find in a specific organism. If this was quite it. I don't know, is there any other comment you would want to add on that regarding the housekeeping genes removal?
Yeah, actually, we are not removing these housekeeping genes. We are storing them in the raw data, raw count matrix, because even if you are working with Seurat or ScanPy, these are two different frameworks, which is one from the R and one from the Python ecosystem. This bot framework first stores the raw count matrix inside their some assays. Then they are also storing the normalized count matrix in another one, and they're also storing the hide variable chains, which will be responsible for the further steps in the downstream analysis in analysis. So we will use these hide variable genes to find out the clusters or to do the PCA. And whether for the differential expression analysis, for the gene set enrichment analysis, for the other kind of analysis, we will for sure use the whole data set. I think it's important.
Okay.
But it's still giving the biological information about these cells.
Yeah, all right. Thank you very much for your input on that. I think it clarified a lot of things that even I didn't know about this.
I think we have a question in the chat box.
Okay, then. Oh, yeah, there was someone who was asking. Oh, okay, fine. Which one was this? Are those zero count genes corresponding to cells considered when doing differential gene expression analysis? Well, DEG comparison per gene. I don't think this. Did you fully understand the question, or should I frame it? Okay.
Yeah, it's a differential expression genes comparison, yes. And as far as I know, yes, they are used in the differential expression analysis.
Okay.
But there could be some other, maybe there is some other steps in the differential expression analysis for the zero counts. Because in the log fold change, it might give an infinite fold change between the zero counts and the sum counts.
True, true.
I think that the DSeq2, for example, has something for this, handling for this kind of issue. Log fold shrinkage, as far as I remember. But I'm sure also that in this kind of frameworks, there should be some further steps for them.
All right. There was also a question here before regarding if analysis is performed between clusters, or if it's typically carried out between conditions, like control or disease, as we typically see in bulk RNA sequencing analyses.
Yeah, it's completely up to you, completely up to your story. So if you want to, yeah, actually the clustering based on the expression profiles of the cell types is important, because you have to cluster something, right? You have to have some information to cluster these cells. It could be something different. It could be cell types. It could be, I don't know, the cell states, the disease states. It's completely up to the story. of these experiments. So sometimes you even have the cell types and also some other metadata, such as the disease state. So you can group them according to cell types plus disease state, like the NK risk score high, NK risk score low. It's up to the story.
That extra resolution to really do comparisons between different types of cells. And then, yeah, hopefully, I mean, does anyone else have any questions? I mean, it was quite a heavy first intro for those that weren't quite familiar with the way that single-cell sequencing works, but I'm sure quite a lot of questions may have popped up, like why are we clustering in the first place, and why is it necessary to maybe remove certain things, but yeah. Oh, so these clusters include control and disease samples. Oh, I guess that's what she was asking. Like you have a mix of samples and then you simply cluster them by cell type rather than.
We do this, and then what was the question again? Right.
The initial question was, is differential gene expression analysis typically performed between clusters or between conditions like control and disease?
As you say, it's up to the story.
If you exactly. And she's followed this up with, do these clusters. So I guess what she's implying here is that you have a single sample containing both disease and control, and then you can cluster these. I guess it's not quite making sense with the way I understand the single cell analysis.
Yeah, you can just compare them according to the whole metadata information you have. You can also even artificial label them and you can compare them. It's complex.
That's also, I guess, another question I had before we jump into these other ones here. The current state of the cluster labeling techniques that we have right now. It seems to be largely dependent on what other people have done in the past and then decided to make open to everyone. So how many years do you think it's going to be until we kind of have a comprehensive library of expression signatures per cell? Or
you mean by the like Leiden or Lewin clustering methods?
Yes, more like annotation, cell type annotation libraries that we have at the present moment. Like those are dependent on known cell types.
Yeah,
and they are also subtypes that we aren't quite aware of. So does that particularly excite you in regards to what we may be seeing in the next 4 to 5 years, like maybe new cell types that we didn't think existed or weren't even aware of? Do you think that we could start to see those emerging?
Yeah, then it's really difficult to say something about this because these cell types, yeah, we project in the automatic way. We project into the currently the cell atlas or the databases we had, like the cell types, or Asmus is doing this, but also it's always recommended to do manually annotation at the same time, simultaneously. And because you can just do this by your hand, like that you can manually annotate these cell types, then you can do it by automatic using this azimuth or the cell type is, then you can compare it, and sometimes it's really like concordant results between them. So
okay,
like yes, we are projecting them into these databases and it's increasing, news atlas are coming, etc. But like there could be a different story in your data set, right? It's something possible. So always they recommended to do both strategies simultaneously.
Well, all right. Thank you very much. That was actually quite a solid explanation as well. And then we have another question here from Kubra Çelikbaş, who has asked here if you can explain a bit more about trajectory inference and what it's used for.
Yeah, the trajectory inference is actually looking at the shared, the features between the cells, the near cells, then neighbor cells, then looking at the actually the transformation of these cells into the other stage. You know, like the every cell is not the even in the same cluster, every cell is not expressing the same genes in the same amount. There is some differentiation between these cells, like some cells could be the initial cells in that cell type, and some cells could be immature, some cells could be the mature cell type, like the mature NK cells on immature, like the from stem cells. Let's think about a stem cell. There's a differentiation, and it's just differentiating to another cell type. Like I think let's think about mesenchymal stem cell. It can differentiate into the adipocytes or in another one, and osteoperocyte. And we can understand this lineage, these trajectories between these cell types, between these cell clusters, from immature to mature, or like in the developmental stage, or it can be used in different stories. But mostly for that purpose, we can track the differentiation of these immature cells into the mature cells, or from stem cells to another one. Yeah, for this purpose also we can use the RNA velocity. It's also showing the future cell state. It's a bit different, not the same as the Seurat time analysis, because it's also showing the direction at the beginning. But Seurat time is actually showing the trajectory. It doesn't actually show you the start point of these clusters in unsupervised way. It's both advised to use at the same time, RNA velocity and pseudotime analysis.
Right, I think that was quite a really good comprehensive explanation as well. What you had explained there with the tracking of trajectories from, your immature cell types to your mature cell types. With the data I was working with, they were looking at T regulatory cells and looking at them from the neonatal stage to right up to the mature stage, and trying to maybe get specific markers identified. So you could do pretty much anything with this. So correlate, specific stages with the expression of very specific long non-coding RNAs or microRNAs. And I guess overall, this really just helps us to better understand cell development and cell type differentiation, I guess. Mm-hmm. So it's quite an exciting technology for those of you who are into stem cells, or even if you're not into stem cells, like I just said, but are actively trying to map out and better understand the immune system to target various things like cancer. So,
right.
Okay, and it seems, all right, it doesn't look like we have any other questions here. I mean, yeah, I guess you must have explained everything really, really well if we see these.
Maybe we can just give a quick break, like the 2-3 minutes, then I can grab a cup of coffee or hot tea. And
all right then. I think you definitely do need that. We need you talking for most of the day. So yeah, we can take a 5-minute break.
You don't have to close this session, right? It's
yeah, we're not going to close it. You can simply come back. I think it's 10:59. We can get back. We can get started again between 11:05 and 10 past 11.
Okay, let's just give like 5 minutes break.
All right then. So we'll be back at 11:05 to get the next phase of the workshop started.
Okay.
So I guess everyone else can also grab their tea, water, and yeah, do what they need to do.
See you soon, guys.
See you soon. And again, for those of you who still hadn't joined the RSG or interested in joining the RSG and participating in our community, please don't hesitate to let us know in the chat, and we can get into contact with you and let you know how exactly it is you can join and either be a part of the community by playing an active role, or passively watch and be the first to know when we hold events such as this one. But until 11:05, it's goodbye for now.
That marks the end of our short little break. Welcome back. So I guess for those of you that had missed the beginning of the meeting, we'll just run through a intro of what's going on here and who we have with us. So this is the first day of a hackathon, which is being organized between two groups, the International Society of Computational Biologists, Turkish regional group and Australian regional group. So while we are having our workshop here, the guys over in Australia, I believe, have had theirs 7 hours ago. So yeah, it's the second joint event of the year that is being held between these two organizations. And our main mission is to facilitate the development of students that are interested in bioinformatics and computational biologists by doing events such as this one. And my name is Koray Çavatama. I will be the chair of this particular event for the Turkish side. I'm a molecular biology and genetics undergraduate student at Bursa Uludağ University. And I'm part of the outreach team of Turkey, which particularly specializes in these types of international collaborations. And here with us as our instructor is Ali Yavuz Çakır, who is currently a researcher at the German Cancer Research Institute, which, like we'd outlined before, is a really prestigious place. They've had 2 Nobel Prize winners come out of there, one for discovering human papillomaviruses, and the other one for major developments in microscopy. So once again, thank you for taking your time to come and help us out here, particularly now when you're a little bit sick, feeling under the weather, but still coming to help us and educate us here on single-cell RNA sequencing. So over back to Yavuz Hocam.
Thanks, Koray. It's my pleasure to be here.
Oh, we can't quite get your sound unless that's just a me thing.
How is it now?
Still can't hear you.
I think it's pretty
Is
clear.
anyone else having
I can
audio
hear.
issues from his side?
I think the
Okay. We're all good. I guess that was an issue on my side.
Okay.
Yeah.
So maybe we can move forward with the practical workshop, guys. So let me share my RStudio. By the way, let's ask this again one more time. Are you guys were able to install RStudio and the Seurat library, or is there anyone have some difficulties when installing them? Because you will follow the hackathon today, tomorrow, and Wednesday, and this will be the backbone. This RStudio and this Seurat libraries will be the backbone for your hackathon analysis. So how is it going? Anyone else? Okay, I also assume that it's a big no problem.
Actually, like 4 people reached out me about their problems, and I think they will be able to solve their problems with my answer.
Perfect.
So, yeah.
Perfect. Good to hear this. Thanks, Marika.
You're welcome.
Then let's continue. So you guys can see my RStudio probably.
Yes, we can.
So in this workshop, we will go through with the Seurat framework, single-cell RNA-seq analysis. And in this workshop, actually, as we don't have some specific data, we want to go with some public data sets. And we're planning to use this PBMCA. It's the peripheral blood mononuclear cells public database data sets using multiple sequencing platforms such as SmartSeq and the 10x data. So in your cases, I think in the hackathon, you will use Seurat data, some other 5 different data sets, and I think the Kuray will give more information in the later steps of this, the workshop, maybe at the end of the day or maybe tomorrow. It will be shared with you guys. So but today's we will focus on this public database. Then I first recommend you guys to load the Seurat library and Seurat data library. You can first install them, and I assume that you already did these parts, and you can install the Seurat. Then you can load these libraries into your RStudio. We can just run this code separately. You can type Control Enter, or if you are using MacBook, you can type Command and Enter just to load these libraries into your RStudio. Then, as I said, we will use this PBM CSE dataset, and we will install them. Then, no worries if you cannot catch me for this coding part. I will share this the tutorial with you guys in the GitHub repo. But it would be really better if you, at the same time, simultaneously code with me. Then you can just ask your questions. Then, okay, let's update the Seurat objects. Let's look at it. What's there? So it says Seurat object, guys. You know, like it has already Seurat object, but if you have your own data, or if you download FASTQ files, BAM files, count matrices in your cases, you can just use different functions to input your data into Seurat. For these purposes, I will also show you how you can enjoy with the Seurat codes by looking at the wignets from their websites. But let's just go with these codes, these tutorials, practical workshop at the moment. And I also recommend you that don't focus on the codes, because you definitely can find the codes at somewhere in this web. It's now actually like the trash bins for the information. You can find any information, any codes for the Seurat, for another stuff, or you can just use the ChatGPT, Gemini, Perplexity, whatever you want to use. I think the most important part is the understanding the mechanism, understanding the whole pipeline workflow in this workshop, in this kind of analysis. Otherwise, you can find the codes, but best to understand this pipeline, this workshop, this workflow, then you can just find the codes and you can apply it. So don't focus much about these codes. So let's show what we have in this data set. We actually have data from 5 different sequencing platforms like 10x, Chromium V3, V2. As you may see, these are different. Also the sequencing chemistry, cell-seq, drop-seq, and in-drops. Then in this workshop, I am planning to go with the 10x V3. Then I have to load the count matrix of this data. Then I'm using this code. But if you want to use the others, you can just repeat these code chunks, and you can change this part to other ones. If you want to repeat the analysis, then let's Control Enter. Let's look at the data. So we have a Seurat object, guys. And in this Seurat object, we have nearly 33,000 of features. What we said before in the theoretical background part. Features are, they could be genes or proteins or open chromatin regions or peaks, or it's up to you. It's up to you. These are the features in the data set. In our case, we are working with the transcriptome data, which is a single-cell RNA-seq sequencing data. So the features in our case are the genes. And we have nearly 3K samples. Samples are the cells in our case. So we have 33K genes, so rows, and 3,000 cells on the columns. Then we can also combine the different data sets, but I'm not going to show it right now because if we add more cells, the downstream analysis actually will be more computationally expensive. Then let's go with this data set. So, you know, like you can just Control Enter to display the basic information about this object. And I think one important thing here is understanding the Seurat object. And as I said, I'm going to share you this workshop material. And please check this Ming Tang's YouTube video. He's actually quite famous with this popular the tools investigations and also explaining the basics of this downstream analysis steps. So please just check this out, this video, after this workshop or in the breaks or when you have the time. So Seurat object is a structured container. It's a framework used for storing and also analyzing single-cell RNA-seq data. It's very convenient framework for handling different types of data. It can store row counts, row count metrics. Counts means count metrics, which the rows are genes, columns are the sample cells. And also it can contain the normalized data, the other metadata like the disease state, if a cell has a cancer or cell types or the risk scores or mutation information or whatever you want. It may store dimension reductions, outputs like PCA, UMAPs, clustering results, etc., all in one place. So let's look at the key features of the Seurat object. In the Seurat object, we will first focus on the assay. Assay, I will show you how to reach these assays and the other parts. So this assay includes the row counts. You can access this data by first typing the name of the variable you created for the Seurat object. In our case, it will be pbmc_10x_v3. You can, at the end, you can add this add symbol, then counts to access the row counts. These are the unnormalized count metrics, gene by cell. In the add data section, you can access the log normalized gene expression values. In the scale data section, you can access normalized expression values for downstream dimension reduction. So in the add metadata section, it could have anything else that you want to put there. But basically, it has the number of features, number of counts, and the mitochondrial percentage per cell. These are the important informations, and you can always find them in a metadata section. As I said, this is the number of genes detected per cell. This is an important metric, guys. For the quality control part, we can use this metric. Then we have the total UMI counts per cell. This is the unique molecular identifier. So this is also our count matrix. This is our counts, important. So the percentage of reads mapping to mitochondrial genes. It's very also important quality control metric. As I said, if it's higher mitochondrial percentage, it might indicate the cell is dying or not dying. That could be another story. Always better to be skeptical. Dimensional reduction informations and the edge reductions. It stores the results for PCA, UMAP, TSNA, and also the cell identities. It's storing it says like the cluster cell types, and this is also important. So let's explore the count matrix. In the count matrix, as we always say, the rows represent genes in our case. Columns represent cells, guys. So let's look at them. How are they looking? First thing first, let's check what is this assay, right? So in the assay, we can see the all features and the cells, but of course, it's just showing the first time features here because otherwise, like it's gonna show like maybe the 100 pages of the here, and it's difficult to show it, right? So then let's look at the row counts. We first access the assay, then in assay, we look at the RNA, then you can actually just, maybe I can show it like this. So when you put $, if it's a multimodal data, it can be RNA, protein, attack, or several different things here as an assay. But in our study, we have only transcriptome data. So we only have RNA. And we can just see it by putting $sign at the end of this assay. Also, this is our variable. Let's add the add sign. We can access the metadata, assays, active assay, active idents, graph, neighbors, reductions, whatever you want. It's all stored here. So let's go with the assay, and then let's put the $sign this time, RNA. Then we want to see the counts. So we put the @ sign, so we can see the counts, scale data, normalized data. It's all here, guys. So let's look at the row counts and how is it looking. Let's store it here as a row counts v3 to them. So as you see, this is the dimension of the data. Rows, genes, and these are the number of cells. So let's also visualize how is data looking. So these are let's imagine that these are the columns, these are the rows. Of course, it's not looking that structure weight because we have a limited area here. So how is it looking? Does that mean something to you guys? Anyone wants to comment about this?
I guess I will, but it seems the number of dots here correspond to how many indexes within a specific, I would say, class or section of the Seurat object you're bound to find these counts in.
Yeah, the nice try. But these dots are actually showing that this cell, so for each dot actually corresponding to a cell on the columns, but because of this limited area, we can show these cells as columns. So each dot is a cell actually. So if it's dot, it means that it doesn't have the counts. It's 0. So why it's not saying 0? Because if we put the exact values, exact counts for each cell, for each genes, it means a lot of data in your memory, in your RAM. So it actually consumes a lot of RAM and make things are a bit difficulty on your side in the downstream analysis. So Seurat is basically storing the data, or scMPy, the raw count matrix in the sparse matrix format. In the sparse matrix format, it's just storing the values, the real counts, not the zeros. So, but we can convert it to dense matrix format to visualize the real count matrix. But this is a sparse matrix. And this kind of frameworks with the high dimensions always store their data in the sparse matrix, because there are a lot of sparsity in the data, a lot of zeros. We showed it in the theoretical part, lots of zeros, maybe the 90% sometimes. In ataxic data, it's much more sparsity. So let's convert it to count matrix into the dense format to visualize the real counts. So let's, here, this is, you remember, this is our count matrix in the sparse format. And we use our built-in function as matrix to just convert it to dense matrix, the real count matrix. We did it, then so of course it's a huge data, it's printing kilolots. Stop it. Then let's just visualize some of the genes. Maybe let's like the 50 genes, first 50 genes, you know, like in this slicing, the first part is the rows, the other is the columns. So by saying 1 colon to 50, I say that slice the first 50 genes, then in the column part, only the 5 cells, the first 5 cells. So let's print it. I think even the 50 is too much. Let's make it a bit more compact. 1.5 to 1.5. Yeah. We just visualize the first 5 genes and the first 5 cells. These are in the columns, but again, because of the limited area, it's not looking like the very structured way. So actually, these cells would be here, but because of the limited space, it's just going here. The same genes on the rows and the same counts. So as you may see, we can now visualize the zeros. And as also you may realize that here, there are not like 50s or 100s in this count matrix. But let's just increase it to a 50 if we can capture something like this. Is there any 2, 3, 4 counts? Can you see it? I saw this one, for example. This gene has 5 counts per this cell. So we can just change it. We can look at a specific gene. MALAT1 is, for example, we talk about the housekeeping genes, which is very constantly expressed in all cells. And it's one of them. And then just look at this counts. It's high in the all cells, right? It's not explaining the variance in the data a lot, as you may see. We just visualize 30 cells and the MALAT1 expression of these 30 cells. So let's continue with the quality control step. In this step, we will actually first calculate the common QC metrics, such as the number of features, total counts per cell, and the percentage of mitochondrial genes per cell. So here, actually, we will use percentage feature set function from Seurat. And it will identify the pattern, starts with empty, mitochondrial genes start with this. And the ribosomal genes generally start with RP, RL, or something like this. So let's first use this function to find the mitochondrial percentage and assign it to here, %mito. To them, it's calculating. So let's visualize them. But before visualizing them, we can also look at what's going on there. So these all are, look at the number, 3,200. These are our cells, guys, cell barcodes. Each of them are one cell. And for each cell, we show the percentage of the mitochondrial counts in that cell. For this cell, we have 10% of mitochondrial counts. And for different cell, it could be much higher. So let's visualize how these mitochondrial counts and the other useful metrics are looking in the violin plot. It's very important plotting the function. So let's just do it as a bit. Here we have three metrics, guys. Number of features detected per cell. So a cell could have 200 genes expressing, or 500, or 5,000, or 10,000. So this is an important metric about this cell. If it's an ambient RNA or something like this, we can understand from that parts. So we will use them for the thresholding in the next step. So then this is the total counts per cell, how many counts this cell has, and also the percentage. So in this violin plot, each dot means a cell. And we actually look at the distribution of the number of features across and among the cells in this violin plot. And as you may see in this petty region of this violin plot, we can see that most of the cells are actually fitted there, right? And which means that these are the normal output for these data sets, and some of the cells has a lot of features and some has very low amount of features. Probably these cells are not the real cells or not a quality cells. It could be dying cell, droplets, empty droplets, or whatever. So in this one, we can see the number of counts per cells. So 20,000 counts per cell, for example, or here it's in average, we have like 8,000 counts per cell. Don't forget, each cell is a dot and the percentage of the mitochondrial content. So generally, the average percentage of the mitochondrial counts around like 15%. We can say it according to this violin plot. And there are a lot outliers on the top. It may indicate some biological content. So it's better to check further. But we generally look at this elbow. And generally, as I said, we not include these cells in the downstream analysis. So in the next step, I will actually use this information, like the 15% of the mitochondrial counts, as a threshold to include the cells by looking at their percentage of mitochondrial counts. So here, let's define some filtering thresholds. In the minimum features, I actually wrote here 100, but you can write 200. It's probably more common. Okay, let's just say 200. And max features per cell. It means that a cell should express the gene's maximum amount of 2,500. And if a cell expressing more than 200 and 500 genes, I will filter them. I will not include them in the downstream analysis. Or if it's expressing less than 200 cells, I will not include them. And the maximum mitochondrial counts, we set it to 15%. And if a cell has more than 15% of the mitochondrial counts, I will not include them in the downstream analysis. So, now we can subset our data according to these defined filtering thresholds. Before we go further, let's first look at the data again. Let's print it out. So basically, we have 3,222 cells in our data. So let's look at after filtering how many cells we have. Let's print it out again. As you may see, guys, now we have 2,311 cells. So nearly we lost 1,000 cells in the quality control step. If you increase the thresholds, for example, for mitochondrial one, if you decrease it to 5, I'm pretty sure that you will have just like 50 cells or maybe 10 cells. So it's better to always check these violin plots to put these thresholds for filtering parts. So, we made the filtering. Let's visualize them again with the violin plots. So, let's look at the previous violin plots. Then look at it right now. Because we set the number of features, the maximum number of features to 2,500. So this is our upper limit at the moment. And on the bottom, you can see this, it's around like 400 or 200, sorry. This is our threshold that we set. Number of counts, also what we set. Also percentage of the mitochondrial counts is set to 15%. So we don't have the cells that has more than 50% of the mitochondrial counts at the moment. The data changed a lot, as you may see. So the quality control is important. And don't do the lazy analysis, like just by looking at these WGNets, these tutorials, don't just follow them. It's always important to understand your data, understand your needs, and understand the biology background behind the data set you have, so you can behave more carefully to your data.
So there's a question, like how do we know about the thresholds we should use?
I think this is also quite a tricky question. It's, as I said, it's up to the biology sometimes. Like if a cell type, specific cell type it's known to have more mitochondrial counts. And if you set your threshold to 15%, it doesn't make sense, right? Because you may lose the real cells. But generally, it's used this violin plot to see this elbow, this corner that I showed here, because it's generally show the average mitochondrial content of the cells, and generally thought to be the other cells are the outliers. it might indicate some biological the pathways or dying or under stress. They may just increase the percentage of the mitochondrial counts because it's related with the ATP. Then under the stress, it's just producing more mitochondrial the gene expression. Then it can be in the any stage, like also in the dissociation or in the other steps, or when they take the tissue or the under stress, it's just increase the expression of the mitochondria. Then for this purpose, we generally taught them as an outlier or dying cells. But yeah, sometimes it could be some biological information. Just be skeptical. Like don't follow always just these tutorials thresholds. Like in the thresholds, generally they put it to 10 to 15, but just visualize with the different thresholds. Just visualize your data to understand how is it looking. Compare your data's how is it looking with the other data's, the tutorials data's, to understand further. But there is no actually one question that covers one answer that covers all this the content. So we filtered these low quality cells. Then now let's look at the metadata information we had. This is our variable that we set before. And you remember that we put @ to access the information in this framework, metadata, essays, and the other stuff. Let's look at the metadata. In the metadata, in the rows, we have the cells and the information about these cells as the columns, like the number of counts, number of features, percentage of the mitochondrial counts. Here, the cluster information we have here, but generally, if you are starting from scratch, you will not have this cluster information, guys. This is a public data set which is already annotated, interpreted, and the cell type are assigned already. Then we see this information in the metadata right now, but at the beginning, you will not have this information. So let's go with the normalization after quality control step. We import the data, we do the quality control, then now it's time for the normalization. It's same in the other frameworks. It's always same. Don't forget this order. Then in this normalization step, it accounts for differences in sequencing depth between cells, allowing for more accurate comparison for gene expression levels. Because I showed it before, the gene expression level could be different between cells. Yeah, this is also what we expect, right? But it could be, the variance could be higher. So we have to stabilize it. Then we can do the further principal component analysis and the further analysis. For this purpose, actually, I create some tables from ChatGPT. These are the fictional tables that I created there. But before we go with them, in Seurat, normalization is typically performed using the log normalization, where the row counts are scaled by the total number of counts per cell, then multiplied by a scaling factor. It's typically 10,000. Then log transformed. So we first actually scale the row counts. I will show actually a bit further down how to scale the row counts and multiplying by the scaling factors, then the logarithmic transformation. It's to stabilize the variance and reduce the effect of high abundance genes. So this is from ChatGPT. Let's say that we have a cell, and for this cell, for different genes, For different genes, we have different number of counts, like 50, 200, 1,000, and 0. And in total, we have 1,250 counts per this cell. So we first scale the row counts. Guys, by the way, now I'm actually explaining the background of this normalization step. In the normal Seurat or ScanPy pipeline, you will just type one line of code and it will do all of them. So you don't have to do any mathematics. But I'm just giving you the mathematics of behind this approach of the normalization, because it's a very important step, normalization, also used in the different approach in the bulk RNA-seq, ATAC-seq, or in the many other genomics field. So you don't have to understand it, but I strongly recommend you to understand what's going on in the normalization. So we first scale the gene 1 for cell 1. So gene 1 was 50 counts per cell 1. So the total count was this. So this is the scaled value. We multiply by scaling factor, which we set as the 10,000. This is more common. So now it's 4,000. So we log transform this value. 1 plus 400, it's for the pseudo count. You know, if it's the inputting 0, it doesn't give an error in this way. So it's nearly like 5.99. So let's look at it. For each gene, we look at the row count, scale counts, multiply by scaled size vector, then the log transformed. So at the beginning, actually the values are quite different as we expected, but the variance is too high. Then we actually here we stabilize the values, guys. Look at it. It's actually conserving the structure, but not actually the default change in the log transform way. But it has the structure, like it's still higher for gene 2 from gene 1, right? So this is important, guys, this normalization. So that was the bioinformatics part of this. Then we can just type this code, and this code chunk is actually doing everything easily, quickly, and output. It's the function name is normalize data from Seurat. We input our variable, normalization method is log normalize. You can use something different, but it's the more common. And scale factor is, as I said, 10,000 is the most common. So let's run it. So did it. As at the beginning shared, Seurat object has assays, and in these assays, it stores row counts, normalized data, and scales data. So we first actually look at the count metrics, the row count metrics, unnormalized. Then now we can look at the normalized count metrics. Here we wrote the name of the variable, then assays, then RNA, then counts are the raw data, data are the normalized counts, scaled data is further scaling, and centralized data. And let's look at the normalized data. Oopsie. It's a lot again. Now, actually, let's look at just the head to view for the first 5 cells. And I want to look at the MALAT1 expression level guy. So, do you remember in the dense matrix, I visualized the row counts for the MALAT1 gene. for the first 5 cells, and it was huge. Let's look at it again. This is the row count matrix, as you may see. For each cell, we have more than 200 transcripts, counts. It's huge, right? Because it's a housekeeping gene, as I said. But when we normalize it, the data will look like this. We preserve this structure, like the higher, lower, and it's not that high, the variance in the data right now. So we also finished the normalization at the moment. Now let's go with the feature selection part. In this part, we will look at the highly variable genes we previously talked about in the theoretical part. So not every gene is explaining the data, guys. Like the mallet one is expressed a lot in the every cell. It's not actually indicate some specific particular biological contents in this data. But at the end, we want to cluster these cells. We want to cluster these cells into some biological information, like some cell types, disease states, something like this. So if a gene has a constant expression value across the cells, how can I use it to cluster the cells? I cannot use it. I have to find the highly variable genes. So for this, Seurat is using a method to calculate the variance to mean ratio of each gene's expression across cells. So then these values are then used for the identifying highly variable genes. I also created this formula by the ChatGPT, but you can see the formula. This is the common formula, right? The variance when divided by the total number of cells. Then here we can see the XEG is the expression level of gene G in cell EI, and subtract by the mean of the G expression. But again, like you are not using this mathematics in coding. You already have all the functions in this kind of frameworks. It's just to understand what's going on there. So we will use find variable features function that automatically identifies the highly variable genes features for the principal component analysis or the other downstream analysis. So this is our function. This is our variable. Then we use the VST selection method, and you can give the number of the features that you want to see as highly variable genes. Because when you find the highly variable features, you know, like it's ranked order, then you can ask for the 5,000, 3,000, 1,000. It's up to you. But generally, if we use like the 2,000 to 3,000 or 4,000 highly variable genes explaining the data. So let's run it. So we can access the highly variable genes with this function, variable features, parentheses, our variable. So it's showing all of them. As you might see, it's ranked, it's ordered, guys. It's the first gene. But we just want to see the top 10. So As you see, these are the highly variable genes in the data, which means that it could be expressing like 100 counts in one cell, and 0 in another cell, 5 in another cell, 500 in another cell. It's highly variable. So this is important to find out the some specific information about the clusters in the next steps. Then we don't, but we can scale the data. Again, like the very standardized the expression values across genes and cells. By default, Seurat used highly variable genes identified before. And then also you can regress out by looking at the mitochondrial genes or ribosomal genes or cell cycle effects. So here's our code. These are all genes. As you see, we took the role names from our data. Role names are genes, call names are cells. Then we use scale data function.
By the way, there's a question. How we know like highly expressions genes based on what? Like they just want to know like if you compare something or not.
For the highly variable gene selection.
Yeah.
Yeah, this we can look at the dispersion for the highly variable genes. But for that part, actually, I'm not behaving so skeptical. I generally go with the 2,500 genes, and I actually I'm the ScanPy person, and in ScanPy there are some functions for this, but in Seurat I'm not quite sure. But let me just try to find something to show you guys. Well, I couldn't find it, but maybe in the break I can check it out. If there is any function for this visualization, probably they have. We can just look at this dispersion of the genes
Yeah, okay. Thank you.
there. Now our data is centered and scaled. Then, as you might remember, we can access our data, our matrices, count matrices in this path. Variable add assays, $RNA add counts for row counts, data for normalized data, scaled data for the scaled data. As you might see, now the data is changed a bit more. So now, it's not a graph-based method. It's linear dimensional reduction techniques. Principal component analysis. Guys, our data is it has a lot of information, and it's highly dimensional data. Lots of rows, lots of counts. So how can we, in the next steps, how can we cluster them? Right? How can we visualize them in one plot? It's really difficult, right? So for this, we can use the linear dimension reduction technique. This is used to reduce this complexity of the highly dimensional data by projecting them to lower dimension. And this component analysis identifies the principal components that capture the most variance in the data sets. And these most variance could be A batch effect as well. It could be technical batch effect, biological effect, another biological effect. It's just clustering them. And it's just more compact format. It's allowing us to simplify the data sets while retaining its most informative aspects. So it's quite fast, and that we can identify the principal components that explain the most variance. So, as we said, it's a linear method, and it assumes that the data is linearly separable and works best when the signal in the data is structured. But sometimes these structures, these patterns are nonlinear. So for this, we can use the UMAP. This is the graph-based nonlinear, the dimension reduction methods. But also UMAP requires the PCA. So actually for this, I strongly recommend you guys to watch this video from StatQuest. This guy is amazing for this explaining the statistical, the equations, tools, approach, and this also I think he's a geneticist, because he always give the example with the genetics, RNA-seq transcriptomes, and it's something like this information. So if you have time when we give the break, please, you don't have to watch in the break. You can also watch your time. It's a very nice step-by-step video.
Absolutely agreed. I've used it for lots of pretty much everything I know about machine learning and bioinformatics at this point. Really great guy. He sings songs about all this stuff. It's impossible to not learn. But we do have some questions here that we hadn't addressed. We were kind of just letting you flow.
Yes.
I think every 10 minutes or so, we'll just periodically interrupt you to make sure that these questions don't get ignored. But the first one was if we suggest any other normalization methods other than log transformation. I'm certainly not sure if z-score normalization can be used with single cell data, but
could you please repeat the question?
Anita was asking if you suggest any other normalization methods other than log transformation. So I believe this was log 2, yes?
Well, actually, I have to check this inside the Seurat, but other than Seurat, we use different kind of stuff like the FPKM or TPM, transcript per million. And there are different methods, like I think the FPKM is also quite common. It was more common before the D6 methods and this log transformation. Like in the FPKM, it's called the fragments per the kilobase of the transcript per million map rates. This is also important. It's looking at also the length of the gene that spans the gene, and it's just dividing it to this length, then it's scaling. This is another common method, and I can also suggest TPM. But if you are using Seurat or ScanPy, I think it's better to go with this method, like the normalized method. And I'm also not quite sure if Seurat has the other methods like FPKM.
I have encountered TPM in the few analyses I've done, but it seems that log base 2 transformation is just the way to go, I guess.
Yeah, exactly. It's pretty simple way, and it's much more common than the others.
Yes, thank you very much. Hopefully, and she says that was very insightful. Thank you very much. By the way, I think in future we would also appreciate it, and it would be a lot more interactive if if you're feeling confident enough to directly raise your hand, switch on your mic, and actually ask the instructor here directly. I think that would make this way more of a cool experience. But I do understand the whole anliyorum ama konuşamıyorum thing that most Turkish people have, so not really forcing anyone. But the next question here is, we didn't compare anything, right? Highly expressed based on what? I think this question is to do with the number of counts. It was asked about 10 minutes ago. So
Yes,
at that particular
actually,
point,
he answered that, so we can skip.
oh, okay, good. So I guess that's pretty much it for now. Like I said, I think once every 10 minutes, we'll just, if there are questions, we will occasionally stop Yavuz Hoca if he hasn't responded to them already. And we will also try to respond to any easy questions in the chat. So please feel free to continue, Hoca.
Okay, perfect. Then for the understanding the basics, actually, I will not spend a lot of time for the PCA analysis background. And I, as I said, would strongly recommend you guys to watch this video. And also the StatQuest guy has 2 or 3 different PCA step-by-step, that's a bit like the shorter videos than this one. Yeah, you can also watch them, but please just spend some time, because this is an important the approach in these machine learning algorithms and in this single cell data, RNA-seq data, to understand your data. This is very important, to cluster your data according to their transcription profile. We also use it in the RNA-seq, in the bulk RNA-seq a lot. Then we have this function, it's called run PCA. It's, as you might see, like you don't have to do anything here, like no covariance metrics or no something like this, no mathematics. It's just doing the job, run PCA. Put your variable, then for the features, you know, like as we said, the highly variable genes explaining the data a lot, the most important parts. Then we choose these highly variable genes in the PCA, running the PCA on the top of it. Because the constant genes cannot contribute to the PCA as well a lot. Then we can actually print this PCA, the gene loadings in each PCA, positively or negatively. This is not something that is really important for you, but I also suggest you guys to visualize them. And so, okay, something happened there. I'm gonna check it later. So let's look at the data here. This is also important, which principal components you can use for your downstream analysis, and this is called elbow plots. And there are also different approaches like class 3 or something like this, just to show you which PCAs are more meaningful for picking up for the downstream analysis. So as you might see, we here on the x-axis, we show the principal components, 1, 2, 3, 4, 5 to 20 here. And this elbow plots, it's showing the standard deviation. And nearly at 10 to 12, maybe on the 10, there's an elbow, right? It's just like an elbow. So it's actually showing that after that point, it's the principal components are not explaining a lot about the data. Then for the downstream analysis parts, we can choose the first 10 principal components. But don't do the lazy analysis in your part, in your sections in the feature. Just visualize your data, then you can select what works for you. So by the way, Cry, that when should we give the lunch break?
If we were going to have it at 3, we can give it at any point where you feel like you've got maybe an hour and a half of stuff left. So
Actually,
I think
before we go for the clustering, maybe we can give the break for the lunch because it's more the common and the more connected with the next steps, like the visualization with the UMAP, clustering, then the differential expression parts. I think these are more
okay then.
Then maybe I can share this HTML file with you guys in the Slack channel.
Oh, yes, that would be good because people were asking about it. So I think over lunch, people can take a look at this, maybe try and run the code themselves. I know the PCA is definitely going to destroy some CPUs, but yeah, I think we can take our lunch break. So we'll take an hour-long lunch break. So right now is 12:11. So I guess we'll be back here at 1:15, let's just say. Allow everyone to, grab something to eat, maybe take a look at the code. Feel free to continue asking us questions in the Slack channel. For those of you that aren't in there, you could send your questions to the chats here, but there's no real guarantee that we may see the notifications. Other than that, thank you very much. See you all in an hour at 1:15.
Okay, perfect. See you guys.
All right, welcome back everyone to the second segment of our the first day of our hackathon, which is the workshop. For those of you again who hadn't been able to join when we gave the very first intro and the one after the tea break, this is the first day of a hackathon which has been organized as a joint event between the Australian student group of the International Society for Computational Biologists, whose main goal it is through facilitating events just like this one, symposiums as well, to promote the development and of skills of young computational biologists, particularly students with the student groups. And yeah, this will be the first educational day of a hackathon, which is a competition in the hackathon have already heard the news about what those are going to be. So yeah, hopefully the first segment has been really educational. We have Ali Yavuz Çakır, who's a research scientist at the German Cancer Research Institute, which I think at this point I've explained quite a few times that it's a really big deal. They've pumped out 2 Nobel Prize winners, and he's currently working as a bioinformatician and a researcher there. So thank you very much, and over to you, Ali Yavuz Hocam.
Thanks, Craig, for the quick introduction again. Then I'm actually quickly checking the code so everything is working well. And so you guys had a chance to check these codes. Anyone else? No? The HTML files that we shared. Okay. Thinking that I shared already. Oh gosh. All right. So guys, we basically made the quality control step for our single-cell RNA-seq data. Then we made the normalization on the data and log normalization on them. Then we scaled the data. Then we applied the principal component analysis on top of it. Then now it's time to do the clustering the cells. So let's just do the first go with this. Clustering is the process of grouping cells with the similar gene expression profiles by looking at the transcription expression profiles of each cell. So in single-cell RNA-seq data, clusterings help to identify distinct cell types or disease states or other stuff. It's clustering the cells according to the similarity between these cells. Then Seurat actually uses a graph-based clustering method. It is super effective for high-dimensional data. So this is actually working in two steps. First, finding neighborhoods. It's constructing a nearest neighborhood graph of the cells based on their PCA scores of the cells. Then it's finding the clusters. It's partitions the graph into communities. In our case, it's the clusters using the Lewine algorithm. It's default in Seurat, but Leiden is a better one. It's just published some years ago, and Leiden is like the improved version of the Lewine. And this in the scampi, the default is the Leiden. So first, let's apply this code to our data. First, we will construct this nearest neighbor graph. And we use the find neighbors function from Seurat. Again, we use our data and we are using the first to 10, the principal component. Then, like we look at the elbow plot and we decided it like this is the elbow, the 10th principal component is the elbow. Then in the clustering, we give it, you can also give it like 1 to 20, 1 to 30, it's up to you guys. But the first 10 principal component is explaining our data more than the others. It's more variable. Then we can use the find clusters function with the different resolutions on our data. So here it's creating the number of the nodes and the number of the edges. Nodes in this case is the number of the genes, and the edge means that it's the connections between the genes. It's the constructing the KNN graph. It's from that part. Then it's created the clusters with the resolution of 0.5. What is resolution? Sometimes, actually, let me just focus on in the next part. So first, let's introduce the nonlinear dimension reduction methods such as UMAP and TSNE. So basically, the TSNE was quite popular for like 10 years ago, maybe. But then the publication of the UMAP, actually, it's much more getting popular and reputed than the TSNA. And it's very faster application. And it's preserving the local and global structures. So we can run the run UMAP function from Seurat by using dimensions 1 to 30, 1 to 10, 1 to 20. Here, I just use 1 to 30. It's actually doesn't change it a lot. I think it's what I observe. Yeah, of course, like if you just use the first 2 dimension, it will show much more different results than the 1 to 10 to 1 to 20. But to be honest, like the 1 to 10 to 1 to 15, yeah, of course, this lower dimension, this embedding this UMAP embedding may look different, but the detail, I think it's not that different if you look for some, that has similar dimensions, 1 to 10, 1 to 15. But here I just use the 30 principal components. It's what I use actually in the scampi Python environment. So here we visualize ourselves in a UMAP plot, UMAP embedding. What we see here, each dot is a cell again, guys. Each dot is a cell, and each cells are clusters by looking at their transcription expression profile. So let's say like this cluster 3 and cluster 0 looks like that they have some similar expression profiles, right? Because their lower dimensional space, in this UMAP space, it looks similar, huh? Like this cluster 3 and cluster 0, and maybe this part of cluster 0 to cluster 5. So actually, this is the UMAP space. You don't have to label the cells with the cluster IDs. This is what the data looks like in general in the UMAP space. What we did is, in the previous part, we cluster the cells. Then we label them with these clusters. We created these 10 cluster names, and we just labeled them we just colored them with these cluster IDs. But this clustering is not related and connected directly to this UMAP space. This UMAP space is just a UMAP, all right? Then we are just coloring them with the clustering information. We can color these cells with the many different metadata. With the disease state, it could be like the cancer, and some part could be the normal, or with the risk score high, low. It's up to you. If you have some metadata information for all cells, you can just color them. Here, we use the clustering information. So, as you may see, like this cluster 0, for example, has two different also expression profile, right? It might have. Like this could be maybe a different cluster than this one. It can be. So in the Leiden or the Lewin clustering, we can increase or decrease the resolution. And if you increase the resolution, example like this, so what's it saying? I Okay, we have to run the KNN, then now we increase the resolution, which means that now we have more fine-grained clusters. Did someone ask a question? Oh, Anita has raised a hand. Yes, Anita.
Yeah. Hi.
Hi.
So I'm not too familiar with Seurat, so I just wanted to know if we can customize the K parameter just like in ScanPy to set it to like any given number, for example, 15 or like 20.
For giving this principal components.
Yeah.
We
The
can
K parameter for like the KNN graph.
Yeah. Wait a second. It should be some more function, but for this, actually, I can maybe recommend you to go to the API. Then maybe we can just go together here. But I stop sharing my screen. So, for example, let's go to this function, and we can look at what they have. What
Oh yeah, it's there.
pointers we can. Yeah.
Yeah, thank you.
Good. The number of trees, Euclidean metric, then yeah, if you do the struggle for any part of it, you can just go to the API in the Seurat. It's given in the reference section, and you can just
All right.
go to each function to look at in details, like the top features, top sales for the pre-processing, log normalization. Yeah, there are a lot of functions, I'm pretty sure that you will just use like maybe the 5% of these functions. But you can just go with it. I'm just looking for example, run UMAP. In the UMAP also, there are several parameters you can just customize according to your needs. Yeah.
Yeah, I do just want to point out that this information you've just obtained here can also quite easily be found in the R console by placing a question mark and then the name of a particular function that you're using. If you want to take a quick look at the documentation. So for example, like you were asking, well, what the effect of K parameters would be. If you don't really know if that's there or not, easily question mark, name of the function, and you should be able to see everything there is to see about that particular function.
Exactly. So it's here. Yeah, as also the core I suggest, and you can also just look at it in the R console. But I generally like to see and an overview in the visualized environment, and also with the other functions, and just going to the API or the reference for this.
Yeah, I definitely do agree. I mean, I also didn't know there was a whole website with all the functions written down. I think that's quite good as well.
Mm-hmm. So then, yeah, now what we did, we increased the resolution, guys, which means that it increased the clusters because it's now more fine-grained clusters, much more clusters in data sets. We did it because it was 0.5, now it's 0.7. So let's visualize them again. Okay, run UMAP again. Did it merge? Why did it merge? Maybe it's not increased that much. Yes, as you might see, sometimes in the previous one, I just increased it from 0.5 to 0.7, which is not a huge difference in the resolution, but now I increase it to 1.2. Then you can clearly see that we have more clusters at the moment, guys. It was 10 on the previous one, but now it's 14. So let's give some information, quick information about them. UMAP is faster than the other applications. And because of that, it's often preferred. Previously, it was a TSNE, but now everyone is using UMAP in their papers. It's good for preserving the local and global structures in the data. It allows us to separate the clusters better, like here. But as I previously said, the distance doesn't mean a lot in the UMAPs. It's not a linear dimension reduction method. So it's creating a 2D or 3D map of cells here, 2D, by using the UMAP component 1 and UMAP component 2. So we can also run the other method, this T-distributed stochastic neighborhood emitting, but it takes a bit while. I'm not sure I didn't run it. Okay, it didn't take a lot, but it's definitely slower than the UMAP. So as you see, we have the same clusters because clusters are something different than this UMAP computation or the tsNA computation. It's stored in our data, in the metadata. Let me just show you. So here is the metadata. Then we can look at all the meta informations that we have in our data, like number of counts, number of feature, number of genes, percentage, cluster, cell types, experiment name, etc. So as you might see, these are the cell barcodes, these are the informations, and this is the cluster names, 0, 1, 2, 3. So let's go back then. So this is the other way of the showing our cells in the lower dimension. It's slower. And it might give slightly different results each time. So we can go with actually the differentially expressed features, in our case, differentially expressed genes. If you are working with the ataxic data, it would be differentially expressed, differentially accessible regions. So in this part, we're gonna try to find the differentially expressed genes between the clusters that we created. Helps identify genes that defines the cell types or the states within our data sets. So for this purpose, we're gonna use find markers function from Seurat. It identified genes that are significantly upregulated or downregulated in one clusters when compared with the others, all others, or some specific clusters, such as we can cluster the data, then we can compare the expression profile of two specific clusters. Like we can compare cluster 1 to cluster 3 to see which genes are upregulated in the cluster 1 or which genes are downregulated in the cluster 3. Or we can just compare the cluster 1 against all cells out of the cluster 1. So here I'm sharing one function. This is to find the markers of the cluster 1. So we use the find markers function from Seurat. Then it takes our variable, our data. Then now we want the cluster 1's markers. We indicated here with the ident. So minimum percentage is the as stated here, the fraction of the cells in the clusters that needs to express that gene. So if this gene is expressed less than 25%, it will not account for this gene. So it's also an important parameter here. And yeah, this will return a data frame with the gene names, markers, their fold change between the clusters, and the statistical significance. So let's calculate it. Let's look at the top 5 genes. So these top 5 genes are differentially expressed in cluster 1. It's different than all other cells. This gene is highly expressed, highly upregulated. We call them upregulated in this cluster, in cluster 1. So these metrics are also important. First is the p-value, which shows the significance of these genes, and the average log fold change is showing how many times the fold change between these clusters. Then it's showing the percentage, but it's just a bit the compressed version here. Then also it's showing the post-hoc analysis and p-value, adjusted p-value. I suggest you to mostly focus on the two metrics here, the adjusted p-value, and which is actually averaged from the post-hoc analysis multiple testing of the p-values across the data set. Then the second is the log fold change. This is also the important metric. So if it says 1.8, it means that these genes express 1.8 times more than the others. Of course, this is the log fold change, but we can interpret it like this. If it's like the minus 1 point blah, blah, you can say that it is downregulated in our data set, which means that it has less expression than the other cells. This is the cluster 1 cells. In cluster 1, we have like the 400 cells, and this is the total output. So we can also finding markers for the all clusters. For this, we use the find all markers function. This function compares each cluster to all clusters. Cluster 1 to all cells, cluster 2 to all cells, cluster 3 to all cells. Then finds the upregulated genes for them. So here we use this function. You know, this is the known function parameter that we use, percentage of the expressing these genes in that group of cells, in that clusters. And log fold change threshold 0.25. So if the log fold change is less than 0.25, it will not show it in the data. So let's run it. Yes. So here we have one more column which shows the cluster. So this is the highlighted upregulated genes for cluster 0. And you can just inspect more according to your needs, like for cluster 3. Then let's also do some data wrangling. So here, what we did, we just grouped by the cluster. Then we filtered the data according to the average log fold change, and it has to be greater than 1 this time. But as you also see here, you can add it inside the function as a parameter, or you can just do it separately with the dplyr package. So let's look at the heads again. So then we can visualize the markers for specific clusters, guys. For example, let's create a violin plot that shows the expression level in every cluster. So as you see here, we input 3 different markers, CD3D, CD14, and Spark gene. So as you might see, this CD14 is highly expressed in cluster 7, particularly expressed in that cell. Also, it's expressed in the other cells sometimes, but as you might see, there is no accumulation of the cells because these violin plots is getting fatty when there are accumulation of the cells in that area. But here it's just like a flat line, because there's only one cell here, one cell here, so only two cells express this CD14 in that clusters. That actually means nothing, really. This is biologically interpreting nothing, because in that clusters probably there are more than 100 or maybe 500 cells, and only two cells expressing that gene. It could be even noise, we don't know. So we can focus on that one. This cluster 7 is particularly expressing that gene. This is important, guys, because we have to annotate our the clusters. We previously know the clusters, we created the clusters, but now we want to label them. So we previously said this, we can manually annotate this data. So here actually at some point I shared it. For example, this CD3D is a T cell marker. Yeah, T cells is quite a huge group. But for example, this CD14 is a monocyte marker. So we can say that this cluster 7 is solely expressing this CD14 is probably monocyte, right? Then for sure there are different markers for monocytes. Then it's better to visualize them separately, or just like this. You can just type the markers here, or you can create a dot plot or a heat map just to visualize some top genes in that clusters to understand what is this cluster. Is it a different cell type? It's a cancer cells, blah, blah. It's up to you. And also here, Spark is expressed in this particular, the clusters. Could be maybe the fibroblast. So for this, actually, you have to know some biological information about your data. But of course, you have to know also, this is the other thing. If you are analyzing a data, a single cell data, you cannot always do the lazy analysis, right? You have to know about your data. If you are working with the epithelial cells or PBMC, you should expect something, right, in your data. Then you can visualize your data by giving some prior information. Then you can contribute to the project in that way also. Then, but also you can visualize the top markers in that clusters. So here actually I added here a function. So basically it's just doing some filtering and taking the top, I call it 5, but let's make it 10. So why it didn't work? So, so now we add the 10 markers for each cluster, guys. Then we will do a heat map. Okay, it looks really compressed. Then let's just decrease the resolution a bit to decrease the number of the clusters. So we will run this all markers again. Yes, as you see, we have less resolution right now, less clusters. So also let's decrease it to 5. Yes. Now let's look at this heat map. Is there something here? So on the columns, we have the cluster IDs, 0 to 9. We have 10 clusters, and on the left side, we have, we made the differential express gene analysis. In this analysis, we listed the top regulated genes for each cluster. So here, I just took the 5 top upregulated genes per each cluster. So you can hear the scale for the expression is from 2 to -2. Then identity is the cluster name. Then here, you can see the top genes for cluster 0. It's given by generally the expression profile is higher than the others. And as you might see, these 5 genes, the following ones, are for the cluster 1. It's obvious, right? And it's going like this. For each cluster, we have a set of genes, 5 genes, upregulated. So by looking at these 5 genes, you can actually know about your cell type. What is this GZMH? You can just search this gene, and you can find this information about the cell type. Actually, there are some websites that you can search for these genes to cell type. And just looking at it, in the cell typist website, actually, there is one tool for it, but I can share it later. Then, yes, actually, we went through the quality control of our single cells. We first took our data, we made the quality control, we look at the count matrices, row count matrix, scale count matrix, normalized count matrix. Then we found the highly variable genes. Then we normalize our data. Then we applied the principal component analysis. We applied the UMAP. We applied the clustering analysis. We made the differential expression gene analysis. Then we visualize this data by Weiland plots and heat maps. And guys, I think that it's a bit earlier than the planned one, but it seems like that I'm almost done with this part. But also, I want to show something on the website, but I can actually accept some questions if you guys have. Any question? Okay, I assume that it was a very beneficial one. and then
Yeah, I was going to say we'll definitely interpret that extended silence as a symbol that you explained it all really well.
And actually, I want to show some more things in the websites. But if there is any questions about that part, we can just go back there. So I'm now showing the screen again.
All right then.
So guys, there is today we actually follow the Seurat pipeline, but there are actually two
Seems like we just had a little stint of technical difficulty there. Not too sure.
exactly. Yeah, the internet connection was not stable, just connected to my phone.
The recording is getting long. It could be because of that, but I guess we're fine if we're back.
Okay, I was showing the screen then. So guys, you can use ScMPy or Seurat for your analysis in the downstream part, but I strongly recommend you guys to follow the tutorials given by the tools itself. For each version, there could be some new functions, new parameters, and new added stuff in the guidelines, in the tutorials, vignettes. So you can just go to the Seurat webpage. And you can also go through the paper. So vignettes here. You can read a lot. Everything is here. Everything's about the papers, other stuff. And you in the here on the top, you can see the installation part again. If you struggled, you can just use it as a copy sheet. Then you can follow this get started tutorial. In here, you can see the introductory vignettes, like guided tutorial, multi-model tutorial, if you are working with the ATAC-seq as well, or the protein data like CytSeq. And for visualization, it's a specialized one. There are more functions, some essential command sheets, some transform, data integration tutorials. So if you are integrating different RNA-seq data, for example, you can just follow this one, because it's just starting from the beginning and doing all the analysis. And you can easily repeat this analysis, because today the most important part was, I think, the understanding the core concepts of the single-cell RNA-seq analysis. These quality control, normalization, highly variable selection, and the other parts, clustering, dimension reduction. These are the core concepts. It's same in every framework. But if you want to specialize in your analysis for like the integrating different data sets, working with the multimodal data, or it's completely up to you, up to your needs, and you can just follow one of these tutorials. Integration, reference mapping for multimodal data, or if it's a super massive data like the 5.3 million, you can follow this. And if you have a spatial data, you can also follow here these tutorials. Or if you have the cell hashing data, this is used for the multiplexing cells. You can follow it. There's another specific one for differential expression testing, cell cycle regression. So here you can also see here the same one. So just go there to that website when you stuck with your analysis, also in the hackathon, also in your daily routine analysis. You can just go there and follow one of them. So it's maybe not explaining all the details about some core concepts, but you can just use it like the cheat sheet, right? Just use it. Also, I'm sharing in the chat. Please use it. So yeah, we're all set, huh? It's it's done from the actually the Single-cell RNA workshop part. Then guys, let's make the question answer part. Yes.
Yeah.
Yeah, I think at some point you mentioned that in Seurat the default clustering algorithm is Louvain. I wanted to know if that's fixed or is it possible to like switch to Leiden?
You can. You have to install this Leiden the function, the tool, and after you do this, you can switch onto the Leiden. But the default is, as you said, is Louvain.
I see. Thank you.
Yeah. But in the scampi, it's the Leiden. And also it's more memory efficient. It's Python. So another question, guys.
Mine is more to do with the day-to-day of someone working with single-cell sequencing data. So do you mainly do like everything from testing your workflows on either clusters or servers, or do you ever really feel the need to use your own laptop per se?
Yeah, it's actually the cry. I'm using the cluster nodes because generally the data that we are working on is quite big. Like the latest data I have was around like 100,000 cells for
That's a
the
lot.
modality. It's not only RNA, it's also having the attack data, and attack data is a bit bigger because it's having much more information. It's in the RNA part, you can have the features around like 30,000, 35,000, 36,000, something like this. But in the attack part, it's millions because it's just giving the peaks. It's so sparse. It's super sparse, but it's just giving the peaks. It's yeah. You are doing the peak calling for this purpose.
Okay. And do you recommend maybe after this we learn a lot more of those downstream data preparation techniques? You know, like the preparation of the counts table itself. Is that a very data-intensive task, such that I would need a cluster, or is that something I could easily learn to do on my own local machine?
Yeah, you mean like the more memory consuming the parts?
Yeah, like generally preparing a counts table from like like the sequencing data.
Mm-hmm.
Is that something that you typically do, such that us as students, if we're targeting, if we're aiming to get internships or to do our own research, would that be something we should spend time actually learning? Or would you recommend we focus more on the downstream analyses that you've shown today? Like post this, getting our own education.
I think it's completely up to the laboratory that you applied for.
Okay.
I mean, like the most of the actually the labs are looking for the people that can apply this kind of strategies onto the biological data. So let's assume that there are several wet labs, right? We know this.
Yes.
And these labs they are creating their own data with a single-cell RNA-seq attack, or they can just use the public data, a lot of data. You can create your Atlas scale data at your home. But these people need some bioinformatician that may apply this kind of strategy or this kind of the frameworks, these applications, single-cell applications into this data set. So in this case, you will probably specialize in the interpreting of the biological informations from single-cell data. And also, you will be the expert probably at some point about using different kind of tools.
Okay.
Which tools are working best, which tools are not that good for this kind of data, blah, blah. You can learn this. But this is more like an applied bioinformatician position. But if you apply, for example, to Roberto Patros group from the United States, this group is creating this 11 fry, this aligner, Salmon, Callisto, that kind of the alignment tool. Or if you apply to Stefan the Salzberg from John Hopkins, this guy is creating tools for alignment again for this pre-processing part, for like bow tie, this top hat, cufflinks. That tools are coming from that lab. If you go to that kind of labs, for sure you will not apply these tools to biological information. It's gonna be more like creating tools for this, the processing parts, for these alignment parts, because that lab is specialized in that purpose, right? But if you go to a machine learning lab, probably you will try to create some machine learning application approach for the single cell data, right?
Yes.
It's completely up to the lab that you applied for. So it's really important where you apply it for your PhDs or for your masters, because probably it will be your future.
All right, all right. I think when you talked about machine learning there, you kind of got into like my current interest at the moment, which is not necessarily machine learning, but deep learning and its potential applications to biology in general. Have you at any point in the past felt the need to integrate something like TensorFlow or even something as basic as specific regression types from scikit-learn to use, let's say, alternative classification to what you may find in your average ScanPy or Seurat pipeline?
Yeah, actually, you generally need something like this, especially if you plan to create some tool. You know, like you have to train some data, you have to test them. Then you will definitely apply some machine learning approach. Like the gradient boosting is quite common for this.
All right. Wait, gradients?
Gradient boosting,
Gradient boosting. Okay.
Yeah, this is quite fast and very efficient machine learning algorithm or this kind of stuff like that. You may need to use them, but in the applied bioinformatics positions, generally you don't need to do this.
Okay. Okay.
I don't know, like maybe in the some tools requires, of course, this kind of the approach, but you don't have to know it. You can just, like in the RStudio I show it, you can just use that function. You can just follow this tutorial, but you have to understand the core basics at least, right?
Yeah.
It's like in the applied positions, you don't have to know the machine learning. Generally, you can just apply these tools, but this tool maybe it's machine learning based or deep learning based tools.
All right, then. I think that was quite insightful, I guess. Thank you very much. Do we have any other questions? I thought I'd ask some of mine to, give people time to start thinking about things they may want to ask.
And also here we can see the most of the people will join to the hackathon, right, Koray?
Yeah, I'm definitely seeing some familiar names here. There were also quite a few people, I think, in the initial session who had also not joined the hackathon, but decided to come through, take a look at the workshop, maybe get a glimpse into what this single-cell RNA sequencing we constantly hear being spoken of really is all about. So yeah, I guess I hope that it was productive for those people in particular. But if we don't have any other questions, then I guess we can begin to wrap this up. So hopefully it was a productive and very informative session. It definitely was for me. There were quite a lot of small things I'd never bothered to take a look at that were explained here. Thank you very much for that.
Yeah, it's great.
Yeah, and I think you explained it really quite well. I really did like that initial part where you kind of went a little bit deeper into the types of the different kinds of what can I say, technologies that are used to sequence all of this. In my head, I just have it's all the same thing, no real differences. But you kind of pointed out that there's a certain efficiency advantages that some have over others. But yeah, I guess this marks the end of the first day of the hackathon and the workshop. For those of you who have joined the hackathon, you will be spending the next 24 hours, I believe. Some of you may be working with people from Australia. And yeah, so I guess you guys will be pulling all-nighters. But for those that are working from Turkey, I think you will kind of have it a little bit easier. You'll be able to work during waking Turkish hours. So for those of you that won't be part of the hackathon, essentially, they are going to be, today you kind of got a rough outline of the general uses, but the hackathon is going to be a lot tougher. We expect people to go and apply this to data sets they've never seen before, and also to come up with their own hypotheses. And while the judges, one of which will be Ali Yavuz Çakır, who we've had with us today, and I believe there should be 2 other people, one of which has been the instructor for the Australian side. And I guess I will also plug in and say, if there are any among you who are in the meeting right now who are not a part of the Regional Student Council for Turkey or whatever country that you're in, I highly recommend that you join. You can definitely let us know in the chat, and we can direct you to how you can join and become a part of us, play an active role. We have lots of teams that are helping this really big machine to keep moving. There's certain people that are working with the advertising, people like me that are working with the outreach and trying to make sure that we have, frequent collaborations with our international counterparts. There's article analysis groups, you name it. If you really want to develop yourself as someone who's maybe just interested or really sees a future for themselves in computational biology and bioinformatics, then the RSG is definitely a good place for you to start. Yeah, other than that, thank you very much to Ali Yavuz Çakır again. It was really educational, really informative, and again, really came with a lot of weight, seeing as, you're someone who's had quite a lot of experience in the industry, and you're also working out of one of the biggest powerhouses when it comes to the field of molecular biology. So we really thank you for taking out your time. We'll be seeing you in the hackathon as well.
Hopefully. Thanks for having me, Cry, and thanks
Yeah.
for joining this workshop.
Thank you very much. As for everyone that has joined, thank you all very much for joining as well. It certainly would have been an event to note if you guys didn't join. So we hope to see you in the other events that we'll be holding in future. I'm sure next year we may want to do more hackathons and hopefully one in person. So I think that'll be a lot more fun. It'll be a much better networking experience. And speaking of that, feel free to send messages to people. Get speaking in the Slack channel. That's what that's all about, to kind of get your networking out. And until then, oh yes, I was gonna get to that. Yes, a recording of the session will be made available. So we've been recording now. I think we'll pass it over to our media team, and they will edit it up nice and proper. And then it should be up on YouTube soon. But I think for hackathon participants, they should have access to it via Google Drive. So it'll be the raw thing. I think all 5 hours of the recording, so at least they can kind of get some little explanations if they need to. Are there any questions before we wrap this up? I guess not. Well, thank you all very much for joining. Those that are in the hackathon, see you. Those that aren't, hopefully we see each other in the future. Bye.
See you soon, guys. Ciao, ciao.
See you soon.